Okay, the objective of this study is to increase thermal stability of an enzyme. So to achieve this, let's propose a hypothesis, which is with the introduction of extra intermolecular interactions within protein structure, the protein will become more stable under high temperatures. So do you agree with this? If yes, how do we do this? So what are the methods to achieve the objective, which is to increase thermal stability or thermostability? That means the same thing. First of all, you will need to obtain the full amino acid sequence of the enzyme. You can do this by either running a protein sequencing or even easier to download the sequence of the protein from an online database if the information is available. Apart from this primary structure of the enzyme, you will need to obtain a three-dimensional structure of the enzyme. Why is that so? Because it is the structure of the protein that will determine its function and stability. Then you will need to identify the active sites of this enzyme. Because if you would like to incorporate any modifications to the enzyme, you do not want to touch the active site. Losing the active site, you will lose the enzymatic activity. Similarly, you will like to identify or conserve amino acid sequences. I will tell you what it means later. And now, coming to the important part, you need to identify the flexible regions in the protein, like loops, that makes it less stable, and then replace amino acid in the flexible loops to form new interactions, such as disulfide bonds, salt bridges, and so on. But the question is, how could you change the amino acid sequence? Well, you can do it by mutating the gene that encodes this enzyme. Of course, there are many approaches that you can use. Here is just one example. Okay, this diagram is what we call the multiple sequence alignment. Here you compare the amino acid sequence of the enzyme with similar enzymes from other organisms. Let's say your enzyme is coming from E. coli, then you can compare to similar enzymes from other gram-negative bacteria. So in this diagram, you can identify the active sites of the enzyme, which is a serine in this case, and it also shows you the consensus or the conserved amino acid across different organisms. So if these amino acids are conserved in every organism, it simply means they are important for the function of this enzyme. So you will not mutate the amino acids for the catalytic site as well as the conserved amino acids. This diagram also shows the secondary structure of this enzyme. You can see that they share similar structure such as alpha helices and beta sheets. Okay, so when you have obtained the 3D structure of the protein, you can see that there is a very large flexible loop. And this loop tends to vibrate a lot during high temperature, which will later affect the overall protein structure. To make this loop more rigid, you can mutate two amino acids residues into cysteines that can form a disulfide bond. You expect to see the loop will now come together and become more stable under high temperature. But of course, mutating the amino acid sequences will always come with consequences, such as the change in enzymatic activity in terms of substrate specificity, binding affinities, and so on.